First of all, thanks for the opportunity to present our work. My name is Sriram Vaidyanathan. I'm a postdoctoral scholar in the lab of Dr. Matthew Porteus at Stanford University. And today I'll be presenting our work on developing a gene-corrected autologous airway stem cell therapy to treat CF. I'd like to just start with a brief introduction of how I learned about CF. So I was working on gene therapy technologies for my PhD work. And it was around the time there were reports of this CRISPR-Cas9 technology that enables the precise genetic modification of our DNA. So this was a really exciting development in the field of gene therapies. And so I was interested in seeing if we could use this technology to develop a therapy for single gene disorders. And so I came to the Porteus lab, and it was here I learned about cystic fibrosis. And I did not grow up in a country where cystic fibrosis is that common. And so I'm not trying to reintroduce cystic fibrosis here. So the common consensus seems to be that there are about 70 to 100,000 CF patients worldwide. And most of these patients are thought to be in Europe and North America. But of late, over the last year or two, some of us at Stanford and a few parents of CF children and the CFRI have been interested in the idea that maybe CF is present in other places, particularly in Asia or even in South Asia more specifically. And so if you belong to the demographic, or if you know somebody in that demographic, or you're interested in that topic in general, feel free to reach out to us. The other point I wanted to make here is that, as we all know, CF is an autosomal recessive disease where you have two mutated copies of the CFTR gene, one from each parent. So in the context of a gene therapy, our goal then is to say is that if we even restore one of this copy to a correct CFTR gene, then you might be able to treat that disease. And so that's often our goal as we go through this presentation. Right. The other factor, again, with CF is, as we all know, CF is a systemic disease, and we all know that respiratory complications are currently the major focus of CF, where the lungs show thick mucus and we see repeated respiratory infections. The point I wanted to highlight here is that the sinuses often also present a very similar presentation, where you have thick mucus and inflammation, and you have severe sinus discomfort and infections. And there's also this idea that maybe the sinus acts as this reservoir where drug-resistant pathogens can hide out and seed repeated respiratory infections, which then eventually causes lung failure. And we also all know that small molecule modulators have been a big development in treating CF. And these drugs were approved based on the observation that they significantly reduce pulmonary exacerbations and improve lung function. However, there have been some limitations. So not all patients are eligible to be on modulators. So at present, about 10% of CF patients with certain class 1 mutations cannot be on modulators. We also know that not all patients respond the same way to modulators. So some patients do really well on modulators and some people don't. And then modulators are also associated with complications and contraindications in many people. And the last factor is that modulators still need to be taken every day by patients. So the question then is there room for improvement, and can we develop something that's more durable? And so the answer for this would be gene therapy. And CF has been a target for gene therapy for the last few decades because it's a single gene disorder. Secondly, there is also this perception that the airways are easier to access than certain other internal organs, and therefore it might be a better candidate for gene therapy. And lastly, there's this idea that treating the airways might be sufficient to improve lifespan, at least at present. And Dr. Murray Egan gave a really nice introduction to gene therapy yesterday, so I don't want to repeat all of yesterday's discussion, but the point I wanted to again highlight is that a lot of gene therapy efforts have focused on what I call transient gene therapy. So in this approach, the goal is to express CFTR in the terminally differentiated cells of the epithelium, so the ciliated cells and the mucus-producing cells, and not necessarily target the stem cells. So the terminally differentiated cells over your lifetime get replaced in the order of somewhere between a few weeks to several months. And so as these cells get repeated, as these cells get replaced by new cells, then you might need a second dose of your transient gene therapy. The alternate approach is what I call gene editing stem cells. So here, the idea is if you correct the stem cells directly, then as your epithelium is getting replaced and new cells are generated from the stem cell population, then you get a durable correction of your airway epithelium that would hopefully last the lifetime of the patient. And so most previous attempts at gene therapy have focused on this transient form of gene therapy, where we have tried to express the CFTR gene using various methods. So these have included non-viral lipid-based vectors, or they've included adenovirus or adeno-associated virus-based methods. And unfortunately, these studies did not observe a durable benefit. And some of these challenges included overcoming this inflamed epithelial barrier that's seen in the CF airway epithelia. So people have not given up on this approach. So there are definitely new attempts to deliver better mRNA or DNA into the airway epithelia. And we even discussed some of these trials, especially the RESTORE trial yesterday in Dr. Egan's session. But then our focus is to see if there's a different approach. And so we were interested in this idea that we could take the stem cells of the airway outside the body and then transplant them. So our idea is to get airway stem cells either from the nose and the sinus cavities or from the lungs, and culture them in a dish ex vivo, and then use a CRISPR-based approach to correct these airway stem cells and then transplant these cells back into the patient. The advantage with this approach is that we don't have reason to worry about immune reactions, especially when you use viral agents like adenoviruses or adeno-associated viruses. You worry about immune reactions against these viral vectors. Secondly, we can directly target the stem cells. So when you are delivering your therapy, a viral therapy in vivo, part of the challenge is to navigate this complex epithelial architecture and target the stem cells. But because we can culture the stem cells in a pure manner ex vivo, we can target the stem cells directly. So that's an advantage. And lastly, we can characterize the product well before we transplant it to the patient. So from a safety angle, it's very attractive. The challenge with this approach is that we would need to come up with a method to transplant airway stem cells. So as we started on this approach, we initially decided to focus on CF sinus disease. And we chose this because CF sinus disease affects most CF patients. And as I mentioned before, the sinuses are thought to be a reservoir for pathogens that may be drug resistant. And so this especially becomes important when people undergo a lung transplant. And then secondly, from a safety angle, the sinuses are easy to access and they're less critical organ than the lungs. So when you're trying an experimental therapy, this becomes an important safety feature. And lastly, transplanted patients are sometimes still on modulators just to address their chronic sinus problems. And so for these reasons, we thought sinus disease would be an interesting target initially, and then we could take the lessons we learn from addressing CF sinus disease using this approach and apply that to treat CF lung disease. So our platform briefly involves the isolation of stem cells, airway stem cells, either from the upper airway or from the lower airway, expanding them in vitro, and then editing them using CRISPR-Cas9 and adeno-associated virus vectors, expanding them again, and then embedding them on a biomaterial. And this will be the focus of today's talk. The part that we're still working on is a method to transplant these cells back into patients. So we first started with culturing basal cells for gene editing. And luckily for us, some colleagues at Stanford and elsewhere had figured out methods to isolate airway stem cells and culture them in vitro in a dish. And so initially, we came across work from our colleagues where they had learned how to culture airway stem cell organoids from the lungs. And we were able to apply this method to culture airway stem cell organoids from the sinuses in the nose. And so you can see here that you have stem cell organoids that were nicely expanded using this method. And then if you see the video here, these were then even be able to differentiate, and you can see the cilia beating in these cells after differentiation. And so our pipeline briefly involves isolation of airway stem cells from tissue. So when we first isolate these stem cells, about 15% of the cells we isolate are stem cells that express cytokeratin 5, which is a marker for airway stem cells. And then we culture these cells under defined conditions where we treat them with epidermal growth factor and some other factors that promote the expansion of stem cells at the expense of the other cells. And so after 5 days in culture using our system, we get a population that's 80% pure stem cells. And so we can really robustly expand airway stem cells in a dish at this point from tissue. So then the next goal was to edit these sinus airway stem cells. So before we go too much further, I wanted to introduce everybody to the concept of genome editing. So Cas9 is a bacterial protein that was first reported around 2012, and it was shown that it can edit the mammalian genome. And so Cas9 binds with a piece of RNA called a guide RNA. And the guide RNA is what enables the precision. So the guide guides the Cas9 to a locus. So in this case, it guides the protein to the cystic fibrosis transmembrane receptor locus. And it induces what is called a double-stranded break. And then our gene correction relies on the repair of this double-stranded break. So the cell can repair this double-stranded break using two pathways. So one pathway is called non-homologous end joining. I call this the lazy pathway, where the cell assumes that no information is lost. So it sees a break and it says, I need to repair this break. And it assumes no information is lost, but this assumption is often wrong. And so you either lose information or you gain information, and what this results in is a gene knockout. So we're not interested in this pathway. The pathway we're interested in is called homologous recombination, where if you provide the cell with a template that resembles the cut site, so you have homology arms that really resemble the cut site, and then you can add exogenous information between these homology arms. So you're almost tricking the cell to saying that, oh, I see a break, let me go find a template that I can use to repair this, except we have snuck in new information in this template. And so that's how we can correct mutations or add an extra copy of the CFTR gene. And so this is just a video that demonstrates it further. So you have a cut, and then the cut is being repaired using this template, and this exogenous information that you see in the orange frame here is inserted into the genome, right? And so this had been attempted already for CFTR, and most of this work focused either on intestinal cells or induced pluripotent stem cells. So the first work came from the Netherlands, from Hans Cleaver's group, where they corrected the delta F508 mutation in intestinal organoids. And so this was really exciting, and proof of concept, it showed that CFTR function can be restored. However, the level of correction was rather low, was 0.2 to 0.4%. And secondly, they were using a puromycin-based selection method to boost the survival of their corrected cells, which is not really ideal when you're trying to make a clinical product. The second set of papers tried to correct CFTR using induced pluripotent stem cells. So the idea then is you would correct these induced pluripotent stem cells, and then you would generate airway cells out of those. But the generation of airway cells from iPSCs is still a work in progress. And secondly, they also use some of these selection methods, and there was a need to further improve the correction levels in the CFTR locus. So our question when we started this is, how do we correct airway stem cells directly, and how do we get to higher levels of correction? And so when we started this project, we decided to use a two-pronged approach. So the first approach was to correct the delta F508 mutation in exon 11, because it affects most CF patients. And because it's a small region of the genome to correct, we thought we'll get a higher level of efficiency. And then the second strategy is what we call the universal strategy, where we want to insert the CFTR coding sequence into the beginning of the CFTR gene. And so that way you can restore CFTR function in patients affected by any mutation downstream of the gene. And so when we first started, our challenge was to find a way to modify airway stem cells without killing them. So the way we deliver Cas9 into stem cells is using a process called electroporation. So we literally shock these cells in order for them to take up these correction reagents. And our lab had a lot of experience doing this in blood stem cells. And so the first attempt was to use the blood stem cell protocols to see if we can modify these airway stem cells. And we could see a little bit of indels, but we could actually never see any correction of the CFTR gene when we used this approach. And importantly, most of the cells just died. And so the first part of the project was really to figure out how do you keep the cells alive when you modify them. And so briefly, what we did was we figured out that if you use this reagent called OptiMEM, when we electroporate these cells, you boost the survival of these cells and you improve the efficacy of the editing events. Secondly, we also learned that we had to add ROCK inhibitor to improve the cell survival after the electroporation process. And then we screened a variety of adeno-associated viral vectors, and we found a serotype that transfects airway stem cells efficiently. And we also identified a dose range that was effective when used in airway stem cells. And so together, using all these four improvements, we then again were able to modify the F508 mutation. And so we were able to correct that F508 mutation in airway stem cells obtained from 10 different CF patients. Most of these samples were obtained from the upper airway or the sinus. And then we also tested this approach in some bronchial derived airway stem cells. And we saw somewhere between 40 to 60% correction of the F508 mutation in these samples. So then the question is, okay, we corrected this mutation, but did we actually restore CFTR function? So we took these corrected stem cells and then we differentiated them using an air-liquid interface. So this interface mimics the physiology that you see in your airway, where your cells are exposed to tissue on one side and air on the other side. And so then this prompts the stem cells to undergo differentiation. And we were able to see a fully differentiated airway with ciliated cells, with cilia beating and mucus producing cells as well. And then we put these cells in an Using chamber, and this was work done in collaboration with Zach Seller's lab at Stanford. And so the differentiated airway epithelial sheets went into what's called an Using chamber. And this chamber has a chloride gradient. So on one side, you have a high chloride amount of chloride ions, and the other side, you have low chloride ions. And then we look for the transport of this chloride ion through this gradient, and we measure the movement of this current across the epithelial sheet. And it's a little more sophisticated than that. So we add some agents that open or close these ion channels so that we know for sure that the currents that we're observing come from CFTR. So we first use amiloride to block the activity of ENaC sodium channels that can confound these measurements. And then we add forskolin, which activates the CFTR channels. And then we add CFTR inhibitor, which inhibits CFTR activity. And we look for this drop in CFTR activity when we add CFTR inhibitor to measure how effective our restoration is. Right? So when we take a sample from a CF patient before correction, you see that there's no response to forskolin, and then there's no response to CFTR inhibitor. So it's the same trace here in black. But then when you look at the red sample where we have corrected the delta F508 mutation, you see that there's a restoration of CFTR inhibitor function here. It's less than what you observe in a wild type patient in blue, but it's still better than what was there in the uncorrected sample. And so we did this in 10 different samples, and we saw that we could get somewhere between 20 to 40% restoration of CFTR function in upper airway basal samples. And in lower airway basal samples, we got somewhere between 40 to as much as 80% restoration of CFTR function. And of course, then the question is this therapeutically relevant for getting clinical benefit? So we think it is, because when you look at patients with class 4 or class 5 mutations, they have a better prognosis and a longer survival associated with those mutations. And when studies have looked for the level of CFTR activity in these patient samples, they have seen somewhere between 2 to 15%, either when you look at patch clamp or when you look at sweat chloride. And so we think our 40 to 60% restoration of CFTR function is really relevant when you look at the delta F508 approach. But as we all know, there are about 1,700 different mutations that have been reported in CFTR with varying degrees of severity, like we all know, right? So the class 1 mutations are the most severe, which don't have a therapy at present. And we have other mutations which have some therapeutic options at least, right? So the question is, can we correct all these other mutations? And if so, do we need 1,700 different products to go after each mutation? And so to overcome this idea of having to come up with 1,700 different products, we came up with this universal strategy. So the idea in the universal strategy is you add the coding sequence of the CFTR gene in exon 1. And so this can restore CFTR function regardless of the causative mutation and where it occurs in the gene. And so it can correct any mutation in the coding region, but not the promoter. So there's less than 0.5% patients who have a promoter mutation on both alleles. So unfortunately, this won't work for them, but it will work for over 99.5% of patients who have mutations elsewhere in the gene. And it would especially be an interesting option for patients with nonsense mutations who don't express any CFTR. So the challenge with the strategy is that CFTR is a large gene, and we discussed this earlier in previous talks, and it's close to the packaging capacity of the AAV virus system. So as a result, you're limited in how much homology arm you can package, and you're limited in your ability to add any sort of selection system that would enable you to select the corrected cells. So we used a split strategy where we still induce a break in exon 1, and then we add the first half of the CFTR gene, and then we put in a second cut site at the end. And so you have a second cut that happens, and then we add the second half of the gene, and then we add a selection marker that's truncated CD19. So the CD19 is a protein that's commonly found in blood cells. We have taken out all the signaling domains and other domains from this protein, and what's only left is just the portion that's on the cell surface. And so we use that to select for the corrected cells using flow cytometry. So after correction, this is how the locus looks. So you have the corrected CFTR gene there, and then you have the selection cassette. And so we first tested this strategy in wild type samples. And so these patients don't have CF. And so we can see that it's less efficient. So 1 to 10% of the cells are corrected. And so about 4% of cells were corrected in this particular sample. But we can use this fluorescence-based enrichment method called FACS, or fluorescence-assisted cell sorting, to take these 4% of cells and select for the corrected cells and get a population that's 60 to 80% pure corrected cells. So we, again, tested the sample tested the strategy in samples from CF patients. And we tested this in 11 different samples. And some of these samples included mutations such as 1282X, R553X, and other class 1 mutations that do not respond to modulators. And we also tested this in F508del samples. And we observed somewhere between 60 to 80% corrected cells that were also positive for the stem cell marker cytokeratin 5 using this strategy. Of course, then the question is, did we actually restore CFTR function? So we took these cells and we put them in an air-liquid interface, and we differentiated them. And we again used the Using chamber assay to see if we restored CFTR function. And as you can see here, the CF samples before correction do not respond to forskolin or CFTR inhibitor. But once they go through our correction method, they show a really nice restoration of both forskolin function and CFTR function. And so we tested this in 4 different samples obtained from the sinus and nose of CF patients. And the point that I'd like to highlight here is, the dots in the black on the right side are samples obtained from non-CF donors. And you can see that after correction, we're really observing CFTR function that's at the level of non-CF patients. So we're getting close to 100% restoration of CFTR using this approach, right? So then the question is, okay, does this work in samples with other mutations? And so we again tested several class 1 mutations. And so in this example, again here, this red trace is from a corrected sample, and this black trace is actually from a patient without CFTR, without CF. And you can see that we get really close to wild type levels of restoration of CFTR function using this strategy in this sample. And then the second one is from a 1282X homozygous sample. Again, you see really nice restoration of forskolin and CFTR function. And so the strategy seems to be able to restore CFTR function in samples affected by a variety of mutations. And so here is a summary of the CFTR function we observed in the 11 different, or I think here it's the 7 different bronchial samples that we got from donors. And you can see again that the black dots on the right are show the CFTR function that you observed from wild type non-CF patients. And the blue samples really are at the same level of CFTR function that you observe in these non-CF patients, right? So we are again at a level that's very similar to what we observe in non-CF patients. And so the study so far, we have seen that we can correct almost all CF mutations in patient samples using our universal strategy. And we can restore a level of CFTR function that's somewhere between, on average, 50 to even 100% relative to what's observed in non-CF patients, well above the therapeutic threshold of 15 to 20% CFTR function. Of course, then the question is, how do we transplant these cells back into a patient? So our goal, our current thought process is that we would need to identify a scaffold to transplant these cells, because initially when we just directly tried to administer these cells, when in animal models, when the animals breathed, everything came out. So here we are testing a porcine small intestinal submucosal membrane, which has already been used by our collaborators in the NIAC lab. And they've shown that this improves the remucosalization of wounds in the sinus after surgeries. And so we wanted to test to see if we can grow our corrected cells on these membranes. And indeed, we can grow these cells on the membrane after correction. And we also see that the cells maintain their stem cell phenotype when they're on this membrane. So our current work is to actually test this scaffold and other scaffolds in animal models to see which of these scaffolds enable successful engraftment of the corrected cells in the sinuses. And so today, what we've shown you is that we can isolate airway stem cells from the sinuses and from the bronchi, and we can selectively expand just the stem cells, and that we can correct these stem cells using our CRISPR-Cas9 and AAV-based approach, and we can get high levels of correction. And then we can restore CFTR function using this approach, and we can see CFTR function that's similar to what's observed in non-CF patients. And we can embed these cells on a biomaterial. And so our future goal is to then optimize the transplantation of these corrected cells back into a patient. So and hopefully we can have an update on that next year. And so I'd like to acknowledge the entire team that's been instrumental in this work. I'd like to thank my mentor, Dr. Porteus, for his amazing mentorship, and the rest of the CF team in the Porteus lab for their help with this project. And then this has been a very big collaborative effort, both at Stanford and across other institutions. And the community has been really great in helping us learn all these different methods that were needed to culture cells and look at CFTR function and so on and so forth. And we'd like to thank our funding sources.